Alberta. Our last speaker for today is Dr. Rowan Martindale, and he's going to be giving a closing address to Palaeo 2023. The interesting about her is that I've been trying to get her up here for 3 years. COVID interfered, and then traveling, and we finally got together and brought her up all the way to Texas.
Yeah.
The title of her talk today is Interdisciplinary Records of Extinction and Exceptional Fossil Preservation in Early Jurassic Tinda Lagerstätte from Alberta. When I first found out about this, I was really intrigued about just the sheer level of preservation.
Let's try.
Something like that.
Go first.
Hear more about that today. Dr. Rowan is Associate Professor in the Department of Geological Sciences at the Jackson School of Geosciences, the University of Texas at Austin. Her research involves both field and lab work, from large-scale mapping to thin section analysis of microfossils. The overarching theme of her work is the connection between Earth and life through time. More precisely, understanding ancient Mesozoic and Cenozoic ocean ecosystems and the evolutionary and environmental events that shape them. The research is interdisciplinary, includes paleontology, sedimentology, biology, chemistry, and oceanography. And it focuses on extinction, carbon cycle perpetuations, events such as the oceanic anoxic events and acidification events, also marine ecology and reef systems, the evolution of reef builders, coral photosynthesis, and exceptionally well-preserved fossil deposits, the Lagerstätte. Conservation is involved in also conservation, paleoecology, and geoscience education, with virtual field trips and paleo working. Now you know why I took me 3 years to get here. She gets involved in so much. So thank you. Here we go.
Wonderful. Thank you so much. Thanks for having me. I know I'm the last talk of the day, so I'll try and keep the enthusiasm up. All right, so I want to start by saying the talk that I'm going to give is not just my work, it's the result of a ton of people's efforts, research, publications, and especially I want to highlight the students that have put their efforts into this work. So I'm going to showcase work that was done by former postdocs, former master's students, as well as former undergraduates, and I'll try and highlight them at the appropriate times. So I always like to start out with, why am I doing this? Why do I bother doing paleontology? Apart from it's super fun, we all know that. A lot of what motivates my research is a concern about modern marine communities. So how do we try and preserve what we have in the ocean and make sure it's there for future generations? And so to do this, I go to these past mass extinction events and try and understand what it is about environmental change or what it is about those particular organisms that allowed those groups to persist. And so really what I'm trying to do is understand how marine ecosystems survive catastrophic environmental collapse. And so we'll talk about that today. So at my heart, I'm an Alberta girl. I grew up about, well, what's now 10 minutes south of Calgary. It was more when I was growing up on a farm. And I grew up with the exceptional museums like the Terrell. I definitely remember going to the Glenbow with classes. And so I was privy to all of these exceptional fossils that Alberta has. I've highlighted a couple beautiful examples of some of these fossils that are preserved in ways that we don't normally see in the fossil record. And so I'll talk a lot about exceptional fossilization, or Lagerstätte, for the singular. It's just a German term for an exceptional fossil deposit. Lagerstätten, in Alberta, we have quite a few. We're very lucky. But they not only offer rare glimpses at unique aspects of communities, so things like soft tissue, things like ecology, things like complete specimens, but these are really important in our understanding of ancient life, and they also require some pretty exceptional circumstances. This is not your everyday fossil deposit. They really require something special. I think actually some of the previous talks have done a really nice job of explaining all the different things that go into that. So I'll talk more about how we get those exceptional fossil deposits. So let's rewind about a decade now, which is a little bit horrifying to say. I want to take you to 2013 when we had the really big floods in Calgary. These were devastating in terms of loss of life, loss of property. I'm not going to go into that too much. It was really hard watching this from afar and seeing communities that I'd grown up in literally washed away. But from the perspective of a paleontologist, these floods offered us really unique views of new sites. They cleaned up and opened up new sites and changed old sites. And so there's a number of really new or better or just newly discovered sites all around southern Alberta. And so the site that I'm going to talk about today is one of those sites that had been worked on previously in the 80s and 90s, but this huge flood really cleaned off a lot of the outcrop, ripped up a lot of material that allowed us to find some of the fossils that I don't think we would have found otherwise. So in August 2013, I was part of a group that came up to Alberta. We studied several different sites, and we were in search of an oceanic anoxic event. So I'm going to refer to the Tawarshan oceanic anoxic event, or the early oceanic, or sorry, the early Jurassic oceanic anoxic event. This is a really interesting interval in history where we see large amounts of really organic rich shales deposited in Europe. There's a ton of work done on this particular event in Europe. And as you can see, there's very little done in other parts of the world. This is a relatively recent update in 2013. None of the dots were really here along this, the coast of North and South America. And so we were coming here to try and see if we could find a signature of this extinction and this environmental perturbation over here in Western North America. So this is sort of this is even post-flood. This is still pretty high waters from this little creek that we were on. This group was out here. We were trying to find the geochemical signals of this particular event. I was sort of along for the ride, just like maybe we'll find some cool clams or something, I don't know. And as we started to do this work, we started to uncover some pretty spectacular things. So this is a little claw there. This is a lobster that runs back. I'll show you a better diagram in a second. We started to find fish, as well as some things that we really didn't know what they were, but we knew that they were soft tissue. So this back here, this is muscle tissue, and then this big blob over here is an ink sac. And so we sort of stopped and said, okay, wait, we've got something really special here. We need to, first of all, we need to make sure we got our permits in place, make sure everything's going well, and figure out what's going on. So an alternative title I could use is the new early Jurassic fossil bonanza that we found at Yaha Tinda Ranch. I say new because when I was initially invited, it was pretty new still. It's not really new anymore, but new-ish. And this site can really tell us, we've done a number of different analyses on the fossils from the site, on the rocks from the site. And so we have a really complete picture of what's going on through time at this particular interval. And it can tell us some really interesting things about the exceptional fossils, how they were preserved, what was preserved, and what life was like in this early Jurassic ocean, and what survived this particular event. So what's so special about the early Jurassic? Why were we even there in the first place? Well, this is a time of extensive black shales, as well as Lagerstätte formation. So there's a couple Lagerstätte or exceptional fossil deposits in Europe that all coincide with this big carbon cycle perturbation event. I say carbon cycle perturbation event because I don't really like the term oceanic anoxic event. That sort of implies that the whole ocean's anoxic. It's not, it's just that the oxygen minimum zone expanded a bit and then fringed on the shelf. So this is basically just a environmental perturbation or a climate perturbation, if you prefer. And some of these Lagerstätte are really quite impressive. This is one of my favorite ichthyosaur specimens from Germany. You can see the skin is preserved on this little guy. This is one of the, this particular deposit was one of the first places where Henry de la Beche reconstructed his first paleoecological artistic reconstructions. And so coinciding with this perturbation, this environmental change, is a mass extinction event. And so we were really interested to try and understand what was happening at this particular interval, but from a lens outside of Europe. So much work had been done in Europe. We really wanted to see what was going on outside of Europe. So what's causing this event? Essentially, the environmental changes or the drivers of environmental change are very similar to some of the ones that we see at some of the big 5 mass extinctions, specifically the end Permian and end Jurassic mass extinctions. So like both of those extinctions, we see the eruption of a large igneous province, which is this big blob down here. And so the environmental change that we see is sort of similar to these events, but what we see is survival rather than wholesale extinction. So that's really interesting to us from a sort of survival standpoint. So the large igneous province is called the Karoo-Farrar-Conaika. This is the oceanic anoxic event is defined by this big negative carbon isotope excursion. Depending on whether you stretch it to 900,000 years or 500,000 years, it lines up something along those lines. But in either case, the large igneous province dates line up pretty well with either the minima of the isotope excursion or the onset of the isotope excursion. And so what we see here is that this emplacement of this large igneous province is really coincident with what we see in terms of the collapse of the environmental conditions as well as the extinctions. So I'm going to go through a couple slides to sort of show you what this looks like. I'll sort of try and stick with the highlights because I know we are at the end of the day. But if I go too fast, please ask questions. So anytime you have a large igneous province eruption, so this is not just one volcano, this is the entire western coast of Canada and the US erupting. Anytime you have these large igneous province eruptions, there's going to be a series of knock-on effects around the globe. And so especially we're looking at things like a rise in the atmospheric CO2, so rapid change in the chemistry of the atmosphere. That usually leads to enhanced weathering of the surface of the Earth. It also warms the surface of the Earth. Those things together will often lead to expanded oxygen minimum zones in the ocean, or oceanic anoxia. If you're working on the shelf, that shows up as anoxia. We see some evidence for acidification, which I'll talk about in a second, but essentially all of these things come together to create extinction within our marine community and terrestrial community as well. So to make this a little prettier pictorially, we have our large igneous province erupting, we have our CO2 rising. That leads to climate warming, which causes heat stress in the shallow ocean. As we increase carbon dioxide in the atmosphere, we have ocean acidification. And then that combination of heat stress and enhanced weathering causes this oxygen minimum zone to expand. So if you're living on the shelf over here, if your oxygen minimum zone is down here, you've got enough oxygen to live just fine. Once that expands or becomes more severe, you might get these low oxygen waters sort of impinging on the place that you like to live. So it's this sort of triple threat, or this deadly trio of things causing extinction in the ocean. Here's just another way to look at that. Again, this Karoo-Farrar-Conaika eruption. If you've got a nice open ocean, like we're off the coast of Morocco where I do some of my work, it's not too bad. But if you've got any kind of a restricted basin, it's very easy to start creating these thick black shales, which is what we see in Europe, which is why we get that oceanic anoxic event term. And we also see that a little bit in Alberta. So again, I'm just using these to show that there is evidence of some of these things happening. I'm not going to explain these in too much detail, but essentially from stomatal indices, we can see atmospheric CO2 basically tripling right here at the onset of the Toarcian oceanic anoxic event. Here again is another example of the Toarcian oceanic anoxic event, and here we see a dramatic increase in temperature. We see perturbations of geochemical cycles. So this is osmium isotopes, which are related to how much weathering is going on around the continent. And so we can see elevated osmium isotopes around around that oceanic anoxic event, again, denoted by this negative carbon isotope excursion. It's not just osmium isotopes. We see this enhanced deposition of organic carbon, this negative carbon isotope excursion. We see excursions in molybdenum, strontium, and a number of other chemical cycles. So all this to say is that when this environmental change hit, it throws everything out of whack. And then just to give you an idea, this is truly a global phenomenon. if we're looking at that carbon isotope excursion, these are all the places around the globe where it's been documented. And in the same way, we've got this really high organic carbon black shales documented around the world as well. So more importantly for us, this is a mass extinction. It's not one of the big five. It doesn't rise to the same level as Ralph and Sapkowski's big five mass extinction. But if you're looking at some of the more moderate mass extinctions, which I always think is a funny term, the late Queensbachia and early Toarcian does show up as one of the significant mass extinction events, which is about here in the early Jurassic. In terms of what goes extinct, we see extinctions across ammonites, forams, bivalves, and brachiopods all show a diminution event. So basically they get smaller across this particular event. We see, again, globally, we see fauna turning over or going extinct. Here's an and we also see a collapse of ecosystems like reefs. So in the dotted line here, this is reef volume. It takes an absolute nosedive across this interval. You can see the reefs are just recovering from the end Triassic extinction and get hit again by the Queensbachia Toarcian or Toarcian oceanic anoxic event. There's also some evidence for changes in the ocean pH. So this is borate, and this is reconstructing pH. It's a little bit messy though, but some people have used this as evidence to suggest that there was acidification going on at this time as well. And then this is again work that I've been doing in Morocco. These are healthy carbonate platforms, but we see this colossal collapse in coral fauna. So the colors are just coral morphotypes, but basically a lot of long-lived taxa bite it right at the end of the Queensbachian and early Toarcian. And the groups that evolve following the Toarcian oceanic anoxic event set the stage for corals and reefs for the rest of the Mesozoic. So this is a really big turnover event, even though it's not one of the big five mass extinctions. And again, the highlight here is that pretty much every system is thrown out of whack. And although it's not one of the big five mass extinctions, we do see some similar hallmarks of this particular event. So this is why we're out at Tinda, is that we wanted to find evidence of this event at that site, and we wanted to try and understand what was going on in the North American record of this event. So again, most paleontological data is from Europe or Morocco, and what's going on outside, we were going to Tinda. And again, we went up to a number of different sites, but we ended up spending a lot of time at Tinda because of what we found. So Tinda, I don't need to tell you where Alberta is, but essentially it's here, just west of Sundre. Everybody asks what Tinda means. It means mountain prairie and stony. So we're obviously occupying land. That's the historic home of stony indigenous people as well as other groups. But what we could do is that by putting together the geochemistry, the sedimentology, and the paleontology, as well as the taphonomy, we can really get a whole system understanding of what's going on at this particular event. So again, here's the outcrop. For those of you who are interested in sedimentology, I've got the strat column on the left, looking at it on the right here as well. Essentially, as we go downriver, we go up section. Most of it is calcareous shales or silty calcareous shales or siltstones with a little bit of carbonate mineralization. So even though it looks like there's a lot of change in the strat column, it's really not much. Mostly the biggest change that you see is going from these more well-vinified layers to these more recessive layers down here. So we've got some beautiful ammonites, not quite as beautiful as John's chlorite ammonite. They're beautiful. This is actually an apticus that's preserved, so these are still pretty exceptionally preserved. And the ammonites allow us to tell us where we are in time. So this is work that was originally started by the late Russell Hall at University of Calgary. It's now being worked on by Andrew Carruthers. And so with Russell Hall's notes and Andrew's work, we can pretty nicely identify where we think the stage boundary is, and then where we think we go into the bifront zone, which is supposed to be where this community is starting to recover. So we can add to that the carbon isotope data from the folks at Virginia Tech. So this was their big push was understanding the geochemistry of this site. And so just because this is a lot of information, I'm going to have a blue line that says stage boundary and a red line that says onset of the carbon isotope excursion that's coincident with this Martian oceanic anoxic event. So that'll be throughout my talk, just to help sort of keep an eye on where we are. So if we're on the outcrop, here's the stage boundary, and here's the oceanic anoxic event. So the first thing all the sedimentologists say is that looks condensed. Yeah, it is. We're looking at several meters here, whereas where I work in Morocco, we're looking at 50 meters. So it is a bit condensed, but we're working at pretty high resolution, so we're still getting a reasonably good record. So what are the environmental stresses and how are the organisms responding to them? So if we look at the geochemical side of things, here's our carbon isotope excursion, there's our stage boundary, and our onset of the Taurasian oceanic anoxic event. If we look at the relative proportions of iron within the sediments, we can identify whether the water column was oxic or anoxic. So no oxygen versus oxygen, or oxygen versus no oxygen, sorry, other way around. But you can also look at whether there was free iron within the water column or free sulfur. So ferruginous or euxenic, depending on where this plots. And so based on the work that was done, it looks like pretty much the entire section is anoxic and euxenic. We can add on to this thallium isotopes, which is thought to be a little more sensitive. And that shows that maybe the bottom part of the section is sort of normal ocean, but that we're really getting into expanded bottom water anoxia, no oxygen from sort of this point onward. They also did some osmium isotope geochemistry, which shows really nicely that enhanced continental weathering of this site. The only problem with this is that within some of these completely anoxic intervals, we have beautiful big trace fossils. So burrows that are a couple centimeters wide, which are widely thought to be built by organisms that need a lot of oxygen to produce these burrows. We also get benthic communities like this lobster here. So that's the lobster's tail, it's his body, his head or rostrum, and the claws out front. So we're getting a community that can live on the bottom of the ocean that's burrowing into the sediment. So that's not really anoxic, right? So it's really hard to reconcile that geochemistry with this paleontology. And so we need to sort of get some better insights to what's going on of the paleontology of this site. But wait, I mentioned lobsters. Lobsters aren't usually preserved. Here's another example of the lobster. I talked about the soft tissue. This is actually a squid, so the back end of a squid here. And then fish with really nice articulation and certain details preserved that I'll point out in a second. So again, once we started finding this, we realized we're not looking at a regular fossil deposit. We're looking at exquisite, exceptional conservat Lagerstätte, if you're familiar with the term. So exceptional fossil deposits that are preserving soft tissue. So just to give you an idea of how rare this is, throughout the entirety of the early Jurassic, these are all the Lagerstätten that are reported. So Tinda is the first marine Lagerstätten from the Jurassic of North America, the entirety of the Jurassic, not just the early Jurassic. And it's the only Lagerstätten from the early Jurassic, from either North or South America. So it's incredibly rare, incredibly important. And so we sort of switched gears and started thinking, okay, what can this tell us? Since this is such a unique deposit, what can we do with this material? How did it form? So I'll just walk you through what we found. We've got some beautiful fish. This one actually wasn't collected by us, that was collected previously. Again, Russell Hall had been working on the site for a while. He never called it a Lagerstätten in any of the published works. I've seen his notes. He refers to this as the Little Holzmaden Creek. Holzmaden is the German deposit where that beautiful ichthyosaur was from. So I think he knew. So anyhow, this is Syrinchus, this long snouted fish that we find from this locality. This is one of the fish we collected. While it doesn't look like much, I want to sort of draw your attention to the gill arches, which are preserved here. We do have ichthyosaurs. We don't have anything quite as beautiful as that little ichthyosaur with skin, but we do have vertebrae and rib cages, as well as some paddles from some of the sites, not at Tinda, but actually no, from Tinda as well. We get articulated and disarticulated crinoids. This beautiful calyx was actually collected by Russell Hall back in the 90s. we have these lovely little lobsters. So this is not the same genus. We actually haven't named this genus yet, but to just give you an idea, this is approximately like that. So that's long scissor-like claw is there. The face, for lack of a better term, the rostrum is there. This is the outline of his body, like that. And then the tail is down there. So again, this is for everything that I've looked through. We haven't published on this one yet, but I think this is a named genus. So it's kind of cool. We also have different kinds of lobsters called encynids, rather than being sort of those flat, sort of circular lobsters. These are more sort of true to what most people think of as lobsters. So this is work done by a former undergrad of mine, Brooke Bogan. And we find two different species of these lobsters. So this one here is Encyna pacifica. Again, that's sort of the lobster tail there, the body, the rostrum, and then the claws. You can see some of the legs down there. And this one is Encyna pacifica is notably has this little like parrot-shaped beak. It's really short and snubby. Claw that's shaped like a parrot beak. It has a long neck. And the other species, Tinda Lagerstätte, has this much longer sort of scissor-like claw. So this is sort of what the two of these look like. Who cares about lobsters? Well, these are actually the first complete specimens that have ever been found of Encyna pacifica. So we now have 3 complete corpses. So we can say a lot more about their ecology. Encyna Lagerstätte had been identified based on 1 specimen before. We now have a ton of material. And so we actually had so much material we could do morphometrics on the claws and make some hypotheses about the different ecologies of these 2 different lobsters. And we actually found a couple of them on the same slab. So that actually means that these 2 different lobsters are living together in the same spot. There are 2 other uncynid lobsters, but those are only found in Europe. But again, with having this exceptional preservation, we can now say something about how these lobsters did or did not persist through the Tolartian event, which we couldn't do with a regular deposit. Again, as much as lobster claws are slightly sclerotized, these are still sort of considered soft tissue fossils. We have some shrimp. Well, we have 1 shrimp that we found, and then he fell apart when we tried to collect him. But that's his tail. Little claws are out there, a little hard to see. But as I mentioned, lots of beautiful ammonites, lots of bivalves, which we'll talk about in a second. The pièce de résistance of this deposit are the coleoids. So coleoids are the group that are, octopus, squid, etc. They're everything that's basically not an ammonite or nautilus within that group. This is a modern vampire squid. And so what you're seeing here is that mantle muscle on the back end of the squid. And then again, this big blob in the middle is the egg sac. So really quite exceptional preservation. This is worked on by former master student John Martindale, who's now a postdoc at Caltech. And again, these things are not preserved in a normal fossil deposit, but we have some really exceptional pieces, many with ink sacs preserved, and many with that muscle tissue preserved as well. And you can even see some of the sort of wrinkles and shrinkage as this thing fossilized. So because we can only see these in Lagerstätte deposits, for the first time ever, we could actually say whether or not these vampire squids made it through this event. And so we could actually say with this work that at least the protuthids and the loligocepids made it through this, this particular environmental crisis and persisted into the recovery. So that was kind of cool, something new that we could say with this material. We have a host of other flora and fauna. We've got pieces of wood, we've got trace fossils, we've got some things that are not quite belemnites, but kind of look like belemnites, Atractites, and then brachiopods as well, which I'll come back to. So I mentioned exceptional preservation. We wanted to do more than just document what fossils were there. We also wanted to figure out how they were preserved. So as was alluded to in one of the earlier talks, taphonomy is a really important part of understanding the paleontology of a deposit. It's the lens through which we understand the community. And so what we really wanted to figure out is how are we getting these fossils preserved? They're so rare. We see them in Europe, but we really don't see them anywhere else in the Jurassic or North America. So what's special about Yaha Tinda? Why are we getting these exceptional fossil deposits? This is work done by a former postdoc of mine. Essentially what we did is we took the fossil material that was small enough to fit in an SEM. And what we did is we not only took images of it, we took EDS maps, so energy dispersive X-ray spectroscopy maps. And what it'll show is just the high colors mean high concentration. So silica is high over here in the matrix, which makes sense. It's black shale, luminosilicates in the matrix, makes sense. On the fossil I should point out, map one is here. So the matrix is in that upper left-hand corner, and then the fossil is the rest of the map. So carbon is pretty high. So we've got some carbonaceous material here. But where we really saw things pop was in the calcium and the phosphorus. So this tells us that this lobster claw is preserved as a calcium phosphate mineral, so like apatite or something similar. So that was all well and cool. We sort of mapped out what was where. There's a little bit of calcite, a little bit of carbonaceous material, lots of calcium phosphate. And we looked at one of those vampire squids as well. I'll pick out map number 1 is sort of in the middle of the specimen there. Map number 2 is on the edge. So again, you're seeing some matrix in the upper left-hand corner. And the same story is here as well, that the matrix has high silica, but the fossil itself is really high in calcium and phosphorus. So calcium phosphate is the real reason these fossils are preserved. Here's the kicker. Phosphatization of fossils doesn't occur in an anoxic or euxenic setting. If we look at where we have this exceptional fossil preservation, it's actually occurring throughout our deposit. So actually quite early in the deposit, we even find some of these vampiropods, as well as throughout the section, not really within the meat of this negative carbon isotope excursion, but all the way up to it as well. So that tells us that we're not in a scenario that's fully oxic, probably not, because otherwise scavengers, predators would pull these things apart, they didn't really preserve. So this is sort of what too much oxygen would look like, if you will. Lots of critters on the shelf, but we're not getting exceptional fossilization. If we have extinia, so free sulfur and no oxygen in the water, we wouldn't have anything, we wouldn't have any benthic organism surviving on the shelf, maybe 1 or 2 little things, but we would only have pelagic critters, and we wouldn't be able to preserve anything through fossilization. Pyrotisation, sure. Other modes of fossilization maybe, but not phosphatization. So instead, what we propose is that we're in a situation where we've got, in oxic settings, we get benthic fauna. As that oxygen minimum zone rises and retreats, we get enhancement of the phosphate within the water column. And then as that oxygen minimum zone retreats again, we get fossilization through phosphatization in the in the oxygenated water. So again, when there's oxygen, we get this benthic community. We get fluctuating dysoxia and euxinia that brings phosphate into the water column or mineralizes it from the water. And then as that oxygen minimum zone comes back, we get that fossilization. So it's this sort of fluctuating dysoxia, which is giving us this exceptional preservation here. Okay, so I talked about the soft tissue stuff. What about the bivalves and bracts? I know my Mount Royal friend wants to hear about those. So there's a lot of stuff that we can do with the quote unquote boring clams and brachiopods. Because they're in such an abundance, we can actually use them to try and understand the physiology of the community and say something meaningful statistically. All right, so you can see just how rare some of the soft tissue material is when we compare it to all the brachiopod and bivalve deposits over here. So I'll walk you through some of this. I'm going to show 2,500 data points in a second, so I want to explain what you're going to see before I show it to you. So this is our strat column on the left. So as always, time goes this way. The taxa are different colored dots. On the left, we have the number of bivalves and brachiopods, and on the right is the body size. I'm going to have to move that because otherwise you're not going to be able to see how body size is measured. All right, so body size is the square root of length times height of the shell. And then just so I don't forget to say it, this is a log scale. So on the bottom left, number of bivalves and brachiopods is on the log 10 scale. Body size is on log 2. All right, So here is all of the communities that we find within this 22 meters of section. You'll note that even within the carbon isotope excursion and before the carbon isotope excursion, we have communities that are, 20, 30, 50, 100 individuals on a single bedding plate. So really abundant communities. You can also hopefully, even from the back, tell that the colors down here are different from the colors up here. We actually see wholesale turnover right at this red line, and I'll explain that a bit better in a second. If we look at the size, we also see a pretty drastic change. Down here in the Plainsbachian, we see very large sized clams and oysters, sometimes even hand-sized specimens. And then when we get into the carbon isotope excursion, we're looking at things that are 1 millimeter to 2 millimeters in size. Really, really teeny tiny bivalves. When we look at the clustering of communities, so how similar are communities, we can see how similar those communities are through time. And the big takeaway is the communities that were alive before the onset of the carbon isotope excursion are completely different from the communities after the onset of the carbon isotope excursion. So the statistics is all nice for a paper, but as was mentioned earlier, we want to tell a story. So let's walk through time rather than looking at those plots. So within the Plainsbachian, we have these nice burrowed horizons. That's within this lower part of the section. Again, telling us that we at least have years of oxygenation. If we expand that to include the late Plainsbachian as well as that first stage of the Toarcian, we also have really high abundance shell beds, but they're all really low diversity. There's not a single shell bed that has more than 5 taxa in it. So it's really low diversity communities, but really high abundance. So these things are proliferating, doing really well for at least a number of years. We can go into more detail on this and actually look at the size distribution. And so basically what you can look for is the shape of the histogram that I'm going to show. If it's unimodal or bimodal, it pretty much means that we've got an even distribution of adults, juveniles, and some adolescents, if you will. If we've got a left skewed histogram, it means we have lots of adults, but very few juveniles. And if we have a right skewed histogram, it's the opposite. We have almost no adults, but a ton of juveniles. So in the Plainsbachian and the first stage of the Toarcian, most of our taxa, so this is the oysters, they're sort of bimodal, but again, not too badly distributed. Our clams, again, this nice unimodal size distribution, whether you're in this zone or this zone, and the brachiopods as well are pretty nicely distributed. So this means we've got adults and juveniles. So within those communities, within those shell beds, the organisms are living long enough to reach adulthood, produce juveniles, and survive okay. We also see our benthic fauna, so our crinoids, all of our lobsters. We have exceptional preservation through phosphatization. So again, this is an environment that's at least seeing a significant amount of oxygen. This is also where we have the most diverse lobster community. A lot of the dashed lines are because a lot of these lobster claws are found in float. So it's really hard to figure out where they're from. But all of our lobster communities that we can really easily nail down, most of them are from this lower interval. This is also where we find the majority of our coleoids. So our most diverse coleoid communities are happening before the onset of the carbon isotope excursion. And again, these are all preserved through phosphatization. Once we hit the onset of that carbon isotope excursion, so that minima, we still see a relatively decent community. So we get a lot of these pseudomytaloides, these little bivalves there. We also have some oysters that are living on ammonites. So they're actually growing on the umbo. This is thought to be when the ammonites on the seafloor, the oyster starts to grow on the ammonite, using it almost like a little island. So these are probably benthic communities, but we also find some evidence of these pseudomytaloides being sort of lined up. So this is the big pseudomytaloides, medium size, little, and teeny. And so it's often thought that when you see that sort of succession of those pseudomytaloides, that they might be on an algae or a kelp. And so those might actually be up in the water column rather than on the seafloor. So we're calling them facultatively pseudoplanktonic. Notably, no exceptional preservation within this interval. but we still see a pretty decent unimodal to slightly right skewed size distribution. So these pseudomilloides are doing pretty well, even though they're within that carbon isotope excursion. In the carbon isotope excursion phase 2, we start to see the stress expand. Our community changes again, and we see a lot of these Bocitra bivalves. Bocitra used to be called Posidonia, so that's hence the name Posidonia shale in Germany. And we also get these Melia granella, again, another little wee bivalve. And if we look at the size distributions, these are all strongly skewed in the direction of having a ton of juveniles and very few adults. So this is telling us that these communities are taking advantage of oxygen when they have it, but it's really not long-lived. And we can actually look at some of these juvenile communities. If you get a little fish particulate or fish skull or something, you'll often get these little nests of Melia granella. We can still see some of the juvenile shells, some of the larval shells on some of these juveniles. So these things are lasting weeks, maybe a month tops. We very rarely see anything that's larger than about 2 millimeters in size, tiny. When we go into the interval where we should be seeing recovery, we're actually seeing sustained stress. We do have a few new critters like this Disconiska. We see some clams, oysters, again, living on our ammonites, and these Melia granella persist. But again, they're really small sizes. This community is still incredibly stressed. And if you look at the abundance, this is where we see really low abundance. So in Europe, where we're seeing recovery, in Tinda, we're not. And as we get into this recovery interval, though, we see a few coleoids recovering. So we do start to see some of that exceptional preservation coming back. And so we might be starting to get a little bit more oxygen in our system. But again, it's still pretty stressful. So to summarize that all, pre-oceanic anoxic event, we've got moderate oxygen stress, but the community that's there is doing pretty well. We've got benthic communities, we've got exceptional fossilization, we've got adults, juveniles, everybody's happy, but we probably do still have intervals of anoxia or extinction, especially considering the geochemical record that we have. Once we hit the OAE, and actually throughout the OAE, we see serious oxygen stress. The only taxa that are there are those that can tolerate low oxygen. And even then, they're really not living much past their juvenile phase. So this is an extremely stressful interval for this community. We put some numbers on these groups just to try and get some kind of a sense for how long they might have lived. Melia granella, we're probably looking at weeks of survival. Maybe some of the pseudomilloides would have been 2 years since we're seeing the adult phase of those as well. But really, this is what's really cool about this is that the geochemistry told us one story, and combining that with the paleontology told us a much more dynamic story of what's going on through this event. If we just used the geochemistry, we would have got it wrong. So combining those two fields was really useful in this case. And I just want to highlight that this concept of fluctuating oxia is not all that unique. So we actually get this off the coast of Texas, where large nutrient loads from Mississippi cause seasonal hypoxia along the coastline. And we also see in a number of other places where you can get seasonal upwelling of dysoxic water. This is off the coast of Chile. And then as the world warms, as things get warmer, the oxygen minimum zone has been expanding, and so some projections show expanded oxygen minimum zone. So this concept of a fluctuating oxycline is actually a pretty, we feel like it's a pretty good way to view this particular anoxic event. It's more of a fluctuating anoxic event. So again, this sort of rising and falling of the oxygen minimum zone would allow for us to have a benthic community. It would allow for that exceptional fossilization, but it might also give us a geochemical signal which is suggestive of anoxia or extinction. Okay, so we also wanted to look at how this fits in a global context. I know Alberta is the best, super important, everybody should focus on Alberta, but we also wanted to see if it was kind of similar to some of the other stuff we'd see. So there are 3 Lagerstätten at this particular interval. Tinda, the Posidonia shale over in Germany, excuse me, and the strawberry bake Lagerstätte up in the UK. So 3 that we know of so far, I should say. So we wanted to see if there were similar taxa and similar preservation at these sites. So just to give you a sort of visual of how these look similar or different, we've got ichthyosaurs at all 3 sites. Some of the fish are a little bit different. We did find a couple gainoid scales over in Tinda, but nothing really to write home about. But again, the Cerminkus we find in both sites. We've got these beautiful colioids at both sites. We haven't got any tentacles yet. I'm on the lookout, don't worry. But again, really nice vampire squids preserved in both localities. Things like lobsters, crinoids, belemnites at all these different sites. Again, often with the same genus, but different species. So this is that one of those European unsigned lobsters. You can see that the claw is very distinct from this one, or at least hopefully you can see. This one's got a little boxing club. This guy's got a lance. All right, so we can compare this to the UK. There's a really well-studied site out in Yorkshire, so we just use this as a nice sort of one-to-one comparison. We can see a lot of the same kind of geochemical changes. We actually see a fairly similar pattern in terms of diversity, of high diversity, with this collapse or complete turnover at the onset of the oceanic anoxic event, as well as these proliferations of these really small Melia granella communities, as well as the Vocitra and the pseudomyloides. So those are very similar. There are some differences. There are some taxa that are unique to Canada. And there's also a number of, there's higher diversity in the UK, but that's largely probably a sampling bias. At Tinda, we're working with this much section. In the UK, they've got these beautiful wave cut cliffs. So when I went and saw this, I was very jealous. This is sort of what we're working with here versus what we're working with there. Again, that expanded section, the much longer study, the sites have been studied for decades, allows for a more nuanced view of the extinction. But again, at Tinda, we don't really see that recovery in the same zone that they see it in the UK. So maybe this Tinda site is just overall a little bit more stressed or just doesn't recover as well as the UK. So the biota are similar. The preservation is kind of similar. We do see a little bit of pyrite, but we really wanted to sort of address how similar that preservation was. When I use Tinda, same story is true for the Posidonia shale. Been quarried for centuries, literally there. Beautiful, exceptional preservation. It was thought that there was a lot of pyrite in the Posidonia shale, but again, these communities within the carbon isotope excursion, communities of those tiny Melia granella, Vocitra, pseudomyloides as well. So very comparable communities, all things considered. So we wanted to understand the preservational pathways. We had a good sense for how the Tinda deposit was fossilized, but we didn't really know what was going on with those 2 sites in Europe. And so my PhD students in Jimmy Sinha worked with my postdoc, Drew Vicenti, to create more of these EDS maps for all the different sites. So we took the same material, so fish, vampire squid, and crustaceans, so usually a lobster claw, and we ran the same analyses on them to see if they were preserved in the same way. And so what you're going to see is there'll be like a long string of aluminum maps, a long string of silicon maps. Again, matrix is usually high aluminum and silicon are usually high in matrix. Calcium and phosphorus in Tinda are high, with all of this sort of fossil material popping out with pink and white. Carbon's also high in some cases as well, but you don't really see a ton of pyrite or other material. So that's Tinda. Strawberry Bay is almost identical, high calcium, high phosphate, patches of carbon. You really only see the aluminum and silicon pop when you're in the matrix material. And the Posidonia shale, again, is the same story, high phosphate and calcium. So in addition to Tinda, those other 2 sites that are occurring at the same time are preserved in the same way. So what's cool about this is this tells us that oceanic anoxic event is basically opening a preservational window, kind of like the preservational window of Burgess Shale type deposits. This is opening a window for the preservation of early Mesozoic communities, which is really neat. And the last thing I wanted to point out is, again, Posidonia shale, everybody thought it was preserved as pyrite. We took stuff that we knew for sure was pyrite, looked at it in EDS, and it's yellow calcite. So Posidonia shale maybe doesn't have as much pyrite as was originally thought, which is good for fossil preservation because pyrite kind of blows up after a while. But that was actually a surprising find for us. We thought for sure there was going to be way more pyrite in Posidonia shale. Okay, so I'm a professor down in Texas. I know I'd rather be up in Alberta too. Maybe not in February, but so part of my job is not just science and research. Another part is bringing it to the students that I teach. I also want to point out, nobody understands why I was so excited about that fossil slab, but y'all might. A bunch of really nice bracket problems on that piece too. So my goal is to take scientific discovery into the classroom. And so what I did with this fossil site in Tinda is that I created a board game that would mimic the fossils that were in this site, the processes that they went through as they transformed from organisms to fossils, and how they were collected and brought back to a museum. So this board game goes through all of those things. Students play against one another. They try and protect their fossils from environmental events. Students get real competitive. They're like, they will be feisty, they'll be fighting with each other. And they try and recover a fossil deposit like the one that we have in Canada. We use examples of material that are, well, we've got pieces, not the pieces from Alberta because those came back here, but we've got specimen collections they can look at to see what would be similar or different. And they basically reconstruct a fossil deposit through playing a game. So it's really fun. It takes them about 2 hours, played by over 1,000 students now, so it's really well tested. We surveyed the students to see if they liked it. Overwhelmingly, students liked it. Some students hate labs, and they're always going to hate labs no matter what. But they really enjoyed playing this game. It takes about 1.5 hours to 2 hours, so it fits really nicely into a lab. They felt like they learned something. We tested them using some post-game questions. Oh, I'm sorry, I didn't point out how to read this graph. More purple is more positive, more green is more negative. So these, I learned something from playing the game, is almost 80% positive in the responses. So overwhelmingly positive responses here. Same thing with the student knowledge assessment. These questions, they were supposed to say no. These questions, they were supposed to say yes. And about 80%, over 80% of the students are getting the right answers. We actually did a second study where we looked at, we gave them the same questions before they played the game, and then the same questions after they played the game. And so this 0 line means no change. So if they got the same score before and after, they'd be on this 0 line. Doesn't matter if it was during COVID and they were playing online or if they're playing in person. Overall, these groups showed significant improvement when playing this game, meaning that their scores increased significantly after playing the game in lab. So that's pretty awesome for me. You know, I'm a scientist. I want to test that what I built actually works, and the answer is it does. So we published this work, showed that it can replicate a lab. It can be used instead. We actually published a print and play version for free. Well, thanks to my dean and a lot of work from me and some other folks at UT Austin, we actually have a physical board game for sale now. For those of you in Canada, it is exclusively found at the Toronto Museum. I have 5 copies out there that I'm willing to sign if you want to buy it. It's a little more expensive than I was hoping for, but COVID and supply chain issues has ruined everything for everyone. But I'm really pleased with how it looks. We actually had an in-house paleo artist do an art piece to represent the Yaha Tinda, which is on the box cover as well. All right, I'm done with the sales pitch. I'll just end by saying that the Yaha Tinda site is an exceptional fossil preservation site right here in our backyard. We see similar deposits in Europe, and we see the same kind of taxa, very similar fossilization, fossilization through phosphatization, which does require oxygen. So we have this dynamic oxycline throughout this Toarcian oceanic anoxic event. And I want to just highlight how important it is to synthesize disciplines. We wouldn't have known what we know about the site without the geochemistry And if we just used the geochemistry, we would have had such a nuanced understanding of how the oxygenation of this site proceeded. And then last but not least, gamification can be really fun. I've had a lot of fun building that and that game, and hopefully it's a new way of sort of sharing paleontology. So I do want to thank the Terrell Museum as well as Parks Canada. This Yaha Tinda is actually on Parks land, so it was quite difficult to access. But thanks to everyone who helped us out, as well as our funding sources, which were predominantly NSF grants. And with that, I'm happy to take any questions.